We had said that our immune system works on ready-made principle and not made to order, which implies that our B cells have to make almost unlimited type of antibodies. Come what may, whatever type of antigen they are exposed to, they should be able to respond. The dilemma is we have only about 30,000 genes, and the amount or different types of antigen an animal can possibly encounter is almost unlimited. So our immune system has to make millions of different types of antibodies. How do we solve that problem? Let's look at that. Even in the absence of an antigen, humans can make 10 to the raised power 12 different antibody molecules. As I mentioned earlier, estimated number of human genes is about 30,000. Mammalian immune system has evolved a unique genetic mechanism that enables it to generate an almost unlimited number of different light and heavy chains in a remarkably economical way by joining separate gene segments before transcription. Here I would like to mention that we share this process with mice, for example. Birds, fish, they do it a different way. Even sheep and rabbit have a different way of generating antibody diversity. Anyways, we'll stick to human and mice. So the first evidence for gene rearrangement. Now I would like to emphasize that I'm talking about rearrangement of DNA, not RNA. We have talked about RNA splicing, RNA processing. This is not RNA processing. This is done at the DNA level. The first evidence came in 1990s. Mouse embryo, an immature mouse cell which was not making an Ig molecule, immunoglobulin molecule, and a mouse B cell making an immunoglobulin molecule. DNA was extracted from both these cells and they were digested by restriction enzymes. I hope you remember we have talked about restriction enzymes. Their DNA was run on an agarose gel by electrophoresis, transferred to a membrane, then hybridized by a probe, DNA probe, which contained both V and C regions. Now, if you pay attention to the slide, you will see the cell that was not making immunoglobulin molecules, the C and V regions are on separate DNA fragments. Here you can see, for example, the C fragment, C region, and here's the V region. We have already talked about this, that our immunoglobulins, for example, our antibodies have two domains, the variable domain and the constant domain. So C region is basically the constant domain and V region is basically the variable domain. These two domains are separate. When they were digested, they appeared in separate DNA fragments. However, when the DNA from a mouse cell which was making an Ig molecule, an immunoglobulin molecule, these two regions were present on the same DNA fragment, implying that somehow these fragments have been moved closer together. They have joined together. So now let's look at the molecular mechanisms of how that happens. So first of all, let's see the antibody structure. Our antibodies, for example, here we have the heavy chain that has been depicted here. Heavy chain is made up of 4 different regions. The variable, V variable region, the D diversity region, J joining region, and mu, as you know, this is the mu means IgM, so this is basically part of the constant domain. So the C domain, the constant domain, which is depicted here in green, and the variable domain, which is composed of these 3 parts, V, D, and J. So the way it works is that our genes that code antibodies are fragmented and there's a pool of these genes, and our B cells randomly select certain segments and join them together. For example, here we have for our V region, there are 100 different segments. Out of these V segments, only one will be picked, and that will be picked at random. So any one of these 1, 2, 3, 4 to all the way to 100 will be picked at random, and it will be combined with the D fragments. So here are the V segments, 1, 2, 3, 4, all the way to 100. One of these segments will be picked. It will be joined by one of the D segments. We have 30 D segments all the way from 1 to 30. These D segments will be picked. One of these D segments will be picked. One of the J segments will be picked out of the 6 we have of this J pool. So we have 3 pools with different number of segments. In V we have 100 segments. In D pool we have 30 segments. In J pool we have 6 segments. The one segment out of these 3 pools will be picked at random, and it will be joined with the constant region. So how do we calculate the different possible number of combinations that we can have? So the simple formula is we multiply 100 times 30 times 6, and if we want to consider that there are 6 constant regions, we multiply by 6, we come with a number which is 144,000. So this is just for the heavy chain. If we think about it, we have 2 chains, heavy and light. Light chain, if we say that we have the same type of formula for the light chain, the total number for possible combinations of the light and heavy chains will be 144,000 times 144,000. So this number goes over a billion. So this is one of the mechanisms B cells use to generate diversity. Now I would like to show you how this happens at the DNA and the RNA level. So here, for example, we have the variable region, which is shown here. This variable region, V, D and J's, one of the segments will be taken from these 3 pools, V pool, D pool and the J pool. So when these 3 segments combine, they will first of all combine with the mu segment, which codes for the M molecule. Remember, B cells first of all make IgM molecules. So this rearrangement happens at the B cell DNA level. Once this has happened, a primary transcription will take place and we'll make the primary RNA transcript. This is primary RNA transcript because it still contains introns. These introns have to be removed. As you know, we have talked about RNA splicing. These introns are removed. We have mature messenger RNA which will move to the cytoplasm. It will be translated into a protein. Now I would like to show you this process at a slightly higher magnification. So up till here it is all DNA, and now here the RNA splicing, as I mentioned, here we have the primary transcript. The primary transcript goes through the splicing process, maturation process, and the introns are removed. The mature messenger RNA is produced, which is translated into an antibody. There are other strategies, other mechanisms that also help B cell generate diversity in their antibodies. We are talking about B cells, but I would like to mention that T cells also have similar process of generating diversity in their receptor. We'll talk about T cells later, but I just wanted to mention that here. Also, the hypothetical number 144,000 times 144,000, that is slightly exaggerated. However, there are other mechanisms which also play a very important role in generating B cell diversity of antibodies produced by the B cells. For example, V, J and C segments, they are rearranged imprecisely. Errors at junctions happen and they create new codons, resulting, of course, when codons change, that results in different amino acid changes. This mechanism also enhances diversity of antibodies produced by B cells. After the DNA sequences are cut out and before they are joined, the enzyme that catalyzes this process is not very precise, and it adds or removes nucleotides, again generating more diversity. Additionally, immunoglobulin genes have relatively high mutation rate, which also generates more alleles and ultimately to antibody diversity. So here we have just talked about another very important feature of immune system, which is the diversity. Our immune system is ready to go and fight war for us no matter what the pathogen is. It has all the different types of armies specializing against pretty much all the pathogens that can possibly be encountered by us.